Hello, I'm Jaebeom Kim from Seoul National University. I'm glad to present our new software, Metabuli. Metabuli is a sensitive and specific metagenomic classifier that jointly analyzes both amino acid and DNA sequences. Metabuli is a metagenomic classifier. Then, what is the metagenomic classifier? We can extract DNA reads from any environment such as soil, ocean, human gut and skin, and patient samples. But they are just anonymous. And the task of classifier is to identify the origin organisms of each read. It can be used to explore or compare microbial diversity of various environments. Also, it can be applied to detect pathogens from pathogens acquired from patients or public spaces. Then, how does it identify the origins? It can be done by searching for similar sequences from the reference genomes, and the similarities can be scanned by calculating local alignments, by comparing k-mers, or by utilizing Bruce-Willis transformation and FM index, which is just basically flexible length k-mers. Based on the approaches, we already have good performance in tools, and the list of tools is quite long. Here are some widely used software, and there are many other tools not listed here. However, they can be grouped by the sequence they utilize, DNA or amino acid sequences. DNA-based tools compare leads and reference genomes, and amino acid-based tools compare 6' translations of each lead and protein sequences. And in this project, we compared Metabuli with these 6 tools. However, there is a trade-off between sensitivity and specificity when using only one of the 2 kinds of sequences. Here is a simple example. Corey's sequence is Jush A, Jush A, Jush A, and 2 sequences in database have some point mutations. If we compare the Corey with the reference sequences, no exact matches can be found, and it leads to lower sensitivity. On the other hand, they are all alanine, alanine, alanine in amino acid sequences, so if we compare them in the amino acid level, even though the blue species has closer sequences to the core sequence, but it cannot be resolved, which leads to lower specificity. So we came up with the idea that let's just combine both DNA and amino acid information to achieve both high sensitivity and specificity. By utilizing both sequences, we can compare sequences in 2 steps. First, we can find exact matches at the amino acid level to achieve sensitivity. Second, we can select the closest matches in DNA level using Hamming distances to achieve specificity. Then, how can we do this? Before looking into implementation details, let's see some benchmarks first. To observe the sensitivity-specificity trade-off, we conducted some benchmarks using prokaryote genomes. Query reads are simulated short and long reads, and in the inclusion test, query subspecies are included in the database so classifiers can make subspecies level classifications. So these tests estimated the specificity of the classifications. As you can see here, orange-colored DNA-based tools showed higher precision and a higher recall than the green-colored amino acid-based tools, because DNA-based tools can exploit point mutations to distinguish between the subspecies. And we conducted the opposite situations, opposite benchmark. In the exclusion test, correlated species are not in the database, they are excluded in the data from the database. So tools should make a genus level classification using the sibling species of the correlated species. In this case, amino acid based tools performed better than DNA based tools because they can detect more homologs by leveraging the higher conservation of amino acid sequences. But with the long reads, MM62 and Kaizu showed impaired performance because they were sensitive to frame shifting index of long reads. And we also included Metabuli in these benchmarks, and as you can see, Metabuli ranked high in all the tests regardless of the inclusion or exclusion tests or short or long reads are used. We also conducted similar benchmark using SARS-CoV-2 data. We used RNA sequence data from COVID-19 patients as our core sequences, and in the inclusion test, 5 variant genomes of SARS-CoV-2 are included in the database. So the task was to detect variant-specific reads. Because DNA sequences changes faster, DNA-based tools could find more reads specific to the corporate variants. In the exclusion test, SARS-CoV-2 was excluded from the database, and only SARS-CoV-1 was provided for the database. So the task was to detect SARS-CoV-2 based on the genome of SARS-CoV-1. Due to the higher conservation of amino-acid sequence, amino-acid-based tools could detect more SARS-CoV-2 reads, as you can see here. When we included Metabuli in this benchmark, as you can see, it showed top-level performance in both tests. It classified similar number of variant-specific reads in the inclusion test, and in the exclusion test, it showed record similar to Kaizu, and it didn't make any false positive hits in control samples. Then let's talk about implementation details. To efficiently store both amino acid and DNA sequences, we first designed Metamer. Naive encoding for DNA 24-mer and amino acid 8-mer requires 8 bits at least, but Metamer uses 60 bits in total. Let me show how it works. First, DNA 24-mer is translated to amino acid 8-mer, which is converted to an integer value that takes 36 bits. And from the DNA and amino acid information, codon usage for each amino acid is encoded using 3 bits per amino acid, so it takes 24 bits in total. So a Metamer store DNA 24-mer and amino acid 8-mer using just 60 bits, which is just 2/3 of naive encoding requires. And in the database creation process, we efficiently cover the whole genomes. To extract amino acid information, we first predict genes in the genomes using Prodigal, but including only the coding sequences makes reads from intergenic regions unclassifiable, so we extend the extended ORFs to cover all intergenic regions. And from them, we extract reference metamers and we sort them in a numerical order. Because we translate the whole genome in a single frame, the amino acid length is 1/3 of the whole genome, so the maximum number of extracted metamer is about 1/3 of the genome length, which leads to a small database size. To further reduce the database size, we removed redundant metamers and stored remaining metamers in a compressed way. Like this figure, we removed identical metamers from the same species, remaining only one of them. For the remaining metamers, instead of storing whole values, we stored the first metamer and increments from previous metamer. We sliced the bit expressions of them by 15-bit windows and retained only minimum slices. Here, for the first metamer, only the most right slice have bits turned on, so only that slice are retained. And for the second metamer, all bits are turned off, so just just one slice with orbits turned off or stored, and so on. And the end flag indicates that these slices are from the same value. And for each metamer, the taxonomic information is stored to be used in a classification process. The interesting point of this compression is the compression rate increases as more genomes are used for the database. With more genomes, more metamers will be extracted and each increment will be decreased. So in the best case, one metamer can be stored using just 2 bytes, so the compression rate becomes 4. Then let's talk about classification process. Metabuli first performs 6-frame translation on the correlate to extract query metamers. And the query metamers are sorted and compared to reference metamers in 2 steps. First, for each query metamer, exact amino acid matches were found, and for the matches, the closest matches in DNA level were selected. In this example, the last reference metamer has DNA hem distance of 6 to the query metamer, so it is rejected, and the middle reference metamer is selected. So the selected matches are used to make taxonomic classification. The matches were first merged by each genus, and the merged matches, like this red bar, are compared to correlate to calculate the sequence identity score, which is 0.7 here. But for the genus B, the merged match has the score 0.35, so genus A is selected here, and for the genus A, its matches are separated by the species, and species A has the higher identity score, so species A is selected. And if matches specific to a lower taxon were found, Metabuli make a classification to the lower taxon. Let's see some benchmark results again. The previous benchmarks was quite extreme because all query organisms were included in the database or excluded from database, but the real-world data is mixture of the reads from rare and understudied case. So to make a more realistic benchmark, we used the plant associated dataset of Kami2. But the results were quite the same. Metabuli showed the best sensitivity and good precision here. And we compared the best performing amino acid and DNA based tools and Metabuli. They are true positive classifications. As you can see, Metabuli covered about 90% of their intersection and 80% and 96% of their relative complements. It means that Metabuli successfully covered both DNA-based and amino acid-based classification, which is what we intended in the Metabuli. We also conducted similar analysis using real metagenomes, but because real metagenomes have no ground truth labels, we just compared which leads were classified by each tool. We used human gut metagenome as an example of well-studied environment and marine metagenome as an understood environment. As you can see, Kaizu have larger area than Kraken2, especially in marine metagenome. It is because in an understood environment, the correct reference distance is increased, so amino acid-based tools have its high sensitivity is quite critical. But yeah, Metabuli also here covered both classifications of Kaizen and Kraken2, and also make more classification which are denoted with letter boxes. So we can say the same results as Kami2 is reproduced. So our conclusion is we successfully demonstrated the trade-off between specificity and sensitivity according to the utilized sequence. And we designed the metamer to efficiently store both DNA and amino acid sequences, and based on that metamer, we implemented Metabuli, and it achieved both high specificity and sensitivity. And I want to say there were rather of support from our lab members, especially from the members, these denoted people with name. Dr. Steiniger is just a wonderful PI, and Dr. Mirdita helped a lot to improve the usability of the software, and Jaebeom Kim helped a lot for the visualization of the workflow and the results, and Ellie helped a lot for writing manuscript. And this project is supported by Seoul National University and National Research Foundation and Samsung Foundation. Thank you for listening, and I hope to see you guys in the conference. Thank you.